Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor for today's event, Agena Bioscience, I'd like to welcome you to a novel approach for multiplex ultrasensitive detection of somatic mutations in circulating cell-free tumor and plasma DNA. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. Now I'd like to introduce our presenter for today. She is Divya Neelam, Master of Science, Senior Scientist in Applications and Technology for Agena Bioscience. Welcome, Divya. The presenter ball is yours.
Thank you, Elizabeth, and thank you, everyone, for attending our presentation this morning. For today's talk, I would like to start with an introduction to our company, Agena Bioscience. Then we will get into the need for ultrasensitive detection and the approach that we at Agena take to increase our sensitivity in order to detect extremely low-frequency DNA mutations that may be present in circulating tumor cells. I will then present to you Agena's UltraSeq panel, which is a multiplexed panel designed to detect 26 hotspot mutations. We will complete today's presentation with a look at some validation data, as well as a case study with a collaborator where we have developed patient-specific custom UltraSeq panels. Agena Bioscience was previously the genetic analysis division of Sequenome. As of earlier this year, Agena is a new company headquartered in San Diego. We have offices in Hamburg, Brisbane, and Tokyo, supporting clients in 37 countries. We use multi-TOF mass spectrometry for the precise detection and quantification of nucleic acids. The mass array system has a broad range of applications in detecting SNPs, somatic mutations, methylation profiles, and other biomarkers. It has been successfully implemented in both basic and translational research and is used in clinical labs for lab-developed tests assaying markers involved in pharmacogenomics, hereditary diseases, and somatic mutations. The manufacturing of the instruments, reagents, and the custom services are conducted in an ISO-certified facility. There are several platforms and technologies in the market today, providing the community with many tools to meet their research needs. On one end of the spectrum, real-time PCR assays such as Roche's Cobas for BRAF detection are low cost and have a fast turnaround time. However, they are limited to low complexity assays or single genes. On the other end, we have next-generation sequencing technologies from Illumina and Ion Torrent, where one can generate extremely large amounts of data, entire genomes or exomes, but that comes at the cost of large bioinformatics investment and a lengthy time to data. The Agena mass array system utilizes multiplexing, allowing the detection of targets from multiple genes in a single reaction. The users can build their own custom panels comprising anywhere from the tens to hundreds of mutations quickly in a matter of hours. The biochemistry process is fast and simple, providing results within 10 hours. The software provides data in an easy, user-friendly format and does not require any additional application for analysis. So why is there a need for ultrasensitive detection? There are several publications showing that the mutations such as the T315I in BCR-ABL, T670I in KIT, and the T790M in EGFR confer resistance to tyrosine kinase inhibitors. In these genes, the highly conserved threonine present at the kinase active site is mutated, altering the drug's ability to bind to the kinase domain. For patients with non-small cell lung cancer, tyrosine kinase inhibitor treatment increases their progression-free survival by 1 to 15 months. However, most of the patients eventually develop resistance to their treatment. One of the most common known mechanisms for the acquired resistance is a secondary mutation in EGFR. The T790M mutation has a 30 to 50% frequency in patients who are resistant to EGFR TKIs. As it is not cost-effective, and not to mention distressing for the patient to have multiple lung biopsies, a non-invasive method is required to detect the resistance-conferring mutation. In a study conducted at the Kinki University in Osaka, Japan, Dr. Nishio's group tested plasma DNA from patients with non-small cell lung cancer for the EGFR T790M mutation. Blood samples were obtained from the patients after they had discontinued gefitinib or erlotinib TKI treatment. The group developed a SABR assay, which has a sensitivity of 0.3% on the mass array system to detect the T790M mutation in the plasma samples. So the presence of the mutation was then confirmed by sequencing clones. When patients under 65 years of age who had a partial response to the TKI treatment were grouped according to their mutation status, the progression-free survival of the patients who were positive for T790M was much significantly shorter than those of patients without the T790M mutation. The authors conclude that the SABR method is a feasible means of determining the mutation status of T790M in plasma and can potentially be used to monitor EGFR TKI therapy non-invasively. In the second study I would like to present to you today, Johan de Bono's group from the Institute of Cancer Research in England used Agena's OncoCarta panel to compare the presence of critical hotspot mutations in matched circulating plasma DNA and archival tumor tissue from patients with late-stage advanced solid tumors. The OncoCarta panel from Agena has a sensitivity of around 10% mutation detection with an input of 6,000 copies or 20 nanograms of DNA. From several studies and based on our experience with developing non-invasive fetal aneuploidy detection tests, we have found that there are typically about 1,400 genomic equivalents per mil of plasma in a typical non-gestational woman. From a typical blood draw of 10 mils, we get about 4 mils of plasma, which typically contains around 6,000 copies of DNA. We can assume a comparable quantity of DNA to be present in the blood of early-stage cancer patients. The Bono's group found that the overall median circulating plasma DNA concentration was 17 nanograms per mil, which is almost threefold higher than in normal healthy individuals, suggesting that the plasma DNA in cancer patients was largely derived from the tumor. And from the results, you can see there was a high concordance for critical hotspot mutations in BRAF, MET, AKT, PK3CA in matched circulating plasma DNA and archival tumor tissue. In cases where a mutation was not detected in the plasma, the authors state that the unmatched result could be due to true novel mutations from evolutionary tumor heterogeneity. What this shows us is that you can use blood samples to detect mutations that are also present in the tumor with a limit of detection of 0.2% and avoid any sampling errors. So, to enable further clinical and translational research in the detection of mutations present in circulating cell-free and tumor DNA, Agena has developed a new biochemistry for ultrasensitive mutation detection. The limit of detection of somatic mutations with the current technologies is around 5 to 10%. With our new ultrasensitive methodology, we are able to increase the detection sensitivity to below 1%, which is much higher than any present-day technology, including next-generation sequencing. This lower limit of detection allows you to identify somatic mutations in circulating cell-free tumor DNA, as well as distinguish low-level tumor heterogeneity. We accomplish this with an assay which is not only cost-effective, but also has a quick turnaround time. So now, let's take a look at the methodology of the UltraSeq panel. We start off with a standard multiplexed PCR reaction where we amplify both the wild type and the mutant moieties, followed by a single base extension reaction. In the extension reaction, we use a mutant-specific biotin triptavidin-labeled terminator nucleotide, Magnetic beads are used to capture the mutant alleles, while the wild type analytes are washed away. The products are spotted onto a spectra chip and analyzed by the MALDI-TOF mass array system. In the example spectra you see, we only get a signal or a peak for the mutant allele and not for the wild type. By eliminating the wild type signal, we have been able to increase our sensitivity to 0.1%. We have also included in the panel several controls. We have 5 assays to ensure that bead capture, cleanup, and elution steps are successful. These capture controls can also be used for semi-quantitation to determine the percent mutant allele, as their peak intensity represents a 1% signal. We also have a process control to ensure that DNA was added and the PCR and extension steps were successful. This allows us to differentiate a negative result where no mutation was detected from a failed reaction. So take a look at the spectra on the right. The first spectra is for 5% mutant. The green dots represent the peak for the capture controls. The blue arrow represents the process control, and the red arrow is the mutant signal. So with a 5% mutant sample, you can see that we have a clear signal and a clear peak for the mutant. And our capture controls and process controls are also present. For a wild type sample, there is no peak for the mutant, but we still have the peak for the process control, ensuring us that DNA was actually added and the PCR and extension steps were successful. In a negative control or a non-template control, again, we don't see a peak for the mutant signal, and also the process control also does not show a peak, indicating that DNA was not added and the PCR and extension steps were not successful as expected. And now, let's take a look at the Ultraseq oncogene panel. The Ultraseq oncogene panel comprises of 26 driver mutations in 12 oncogenes commonly observed in neoplastic cells from various tumor types, both solid and liquid tumors. The mutations in this panel are non-synonymous coding mutations from key genes implicated in resistance to tyrosine kinase inhibitors. Hence, we have key mutations from EGFR, KRAS, BRAF, PIK3CA, IDH1 and 2, in addition to others. The Ultraseq biochemistry enables detection of mutations below 1% from plasma, circulating tumor DNA, heterogeneous tumors, as well as longitudinal samples. The panel requires 14 nanograms of DNA per sample, where the custom panel may require much less depending on the number of mutations assayed. For 0.1% mutation detection, we require about 5 to 10,000 copies of input DNA to eliminate any sampling error. To determine the limit of detection of the Ultraseq assay, we performed a paired analysis with ARMS PCR. ARMS PCR is an allele-specific real-time PCR-based technique. A model system was created by an outside laboratory where they mixed a constant number of BRAF V600E mutated alleles with varying amounts of wild-type DNA to generate mutant fractions ranging from 0.1% to 10%. So here we have the results from Ultraseq, and you can see we have excellent detection from 10% all the way down to 0.1%. And in the small graph in the top left, you can see here the black dots represent the 0.2% samples, the green are the 0.1%, and we have the blue dots, which are the control samples or wild type at 0%. And the green dots of 0.1% are clearly distinguishable from the 0% samples. And now here are the results comparing the ARMS PCR with the Ultraseq assay using the BRAF V600E model admixture samples. The samples were run in 3 replicates. The ARMS PCR was performed on 2 different instruments, hence we have ARMS 1 and ARMS 2. So the results in green indicate true positive or true negative results. The ones in yellow required expert manual calling of at least one of the replicates, and they were given half a score. The false negatives are in red, And so as you can see, ARMS PCR has a sensitivity of about 1%, and below which it's unable to pick up all or some of the mutant replicates. Whereas Ultraseq assay has successfully identified every positive admixture sample down to 0.1% and also does not have any false positives. Once we were able to validate Ultraseq on model sample admixtures, we performed a feasibility study using real samples. We obtained 122 cell-free DNA samples isolated from tumor and circulating cell-free DNA. The plasma samples were previously characterized by ARMS, and we also included 60 wild-type genomic DNA samples, 2 positive controls at 1% and 0.5% that we obtained from Horizon Diagnostics, and 3 non-template controls. So here are the results of that study. So the y-axis represents the mean normalized intensity for the Ultraseq assay, which is proportional to the minor allele fraction in the sample. On the x-axis, we have the results grouped into different bins based on the detection. So the first group here is the controls with the 0, 0.5, and 1% samples. Next is the group of mutations that were detected by Ultraseq and were also present in the tumor, but not detected by ARMS. So these are the samples that were missed by ARMS. Then we have the mutations that were detected by both Ultraseq and ARMS and also present in the tumor. Next is a sample where a mutation was detected by both Ultraseq and ARMS, but not present in the tumor. We think this might be due to a technical error. Maybe the tumor section did not contain the mutation. Then we have a mutation that is positive for ARMS, but not detected by Ultraseq, nor was it present in the tumor. We think this may be a false positive due to a chain of custody event, maybe a wrong sample used in the wrong place. Then we have mutations present only in the tumor, but not detected in the plasma by either ARMS or Ultraseq. So this could be due to poor plasma prep or insufficient tumor DNA in the plasma. And finally, we have the last group of samples where a mutation was not found either in the tumor, and neither did Ultraseq or ARMS find the mutation. So essentially, these are the wild-type samples. So overall, Ultraseq detected the presence of the V600E BRAF mutation in 54 out of the 72 known cell-free DNA samples, while ARMS only detected the mutation in 41 of the samples, so about a 57% detection rate for ARMS. So out of the 122 paired tumor and cell-free DNA samples, 48 were negative in both the tissue and all 3 tests. So now if we take a look at only the 72 cell-free DNA samples with known mutation present, 39 of them were detected by both Ultraseq, ARMS, and were also present in the tumor. There were 18 mutations in the tumor that were either not present or detected in the cell-free DNA by either Ultraseq or ARMS. And Ultraseq detected a mutation in 15 samples that were also present in the tumor but were missed by ARMS. There was one sample that was positive for both methods but not present in the tumor. Again, we think this might be due to a technical error, maybe with a tumor dissection. And then there's one mutation that was detected only by ARMS but not present in the tumor or Ultraseq. And this is being investigated for a possible chain of custody event. Now to our case study where we designed and implemented patient-specific custom Ultraseq panels. Tumor heterogeneity is a phenomenon where tumors may comprise of subpopulations of cells with distinct genetic alterations that may lead to different biological behavior. In their mini review paper from 2013, Dr. Swanton and his colleagues surveyed several publications and showed that a systematic approach to the study of molecular heterogeneity in cancer is required. Single biopsy specimens of primary tumors may not fully represent a genetically diverse malignancy with multiple metastatic sites, and the sequencing technologies may not be sufficiently sensitive to detect low-frequency events in tumor subclones. Current clinical practice is guided by the molecular analysis of primary tumors, assuming that all their characteristics are carried over to the metastases later in the disease. So we risk missing potentially important molecular changes if repeat biopsies are not performed during the evolution of the disease. Hence, they recommend longitudinal analysis of the tumor evolution through the course of the disease by acquiring multiple tumor samples from different regions of the primary and metastatic tumor sites, and also at sequential time points during the disease course, including before, during therapy, and at disease progression. As the requirement for multiple tumor biopsies is, again, ethically and clinically challenging, they suggest new approaches such as cell-free circulating tumor DNA from plasma as a prime source for analyzing tumor evolution. So we are currently collaborating with Dr. Swanson's group to determine the feasibility of studying tumor heterogeneity through the analysis of circulating cell-free tumor DNA in plasma. So through this case study, we are looking to establish whether patient-specific driver mutations can be identified in circulating cell-free DNA. And can this be done non-invasively and at a low cost and in a timely manner using custom ultraseq panels. We're also trying to see if we can distinguish the driver mutations which account for the intratumor heterogeneity between the primary tumor and the metastases. So the study comprises of 7 patients, for each of whom a custom ultraseq panel is created based on their tumor profile. Agena offers custom ultraseq panel design through our Assays by Agena service, and we're also soon launching a new software design to enable our customers to design their own ultraseq panels. So we have started the study and so far have the results for 1 lung cancer patient. So we have DNA from a few different sources from this patient, genomic DNA from intertumor regions and the metastatic tumor, as well as DNA from plasma. So we designed a custom ultraseq panel comprising of the driver mutations which were discovered through sequencing the tumor biopsies. So here we have the results from sequencing. So 7 mutations were found in genes such as BRAF, MLL2, TP53. And so here we have the location where these mutations were found. R1 is the right lung tumor. R2 is the second tumor from the right lung. R5 is from the lymph node. So they categorize the mutations based on how important or the confidence level. And so 1 is where the high confidence mutation of present in driver genes with known oncogenic or disruptive mutation in tumor suppressors. 2 are potential driver genes that are known to be cancer-related, and the mutation is likely to be oncogenic or tumor suppressive. 3 is where the consequence of the mutation is unknown, but the genes are known to be cancer-related. And finally, 4 is the category where the mutation and gene are of unknown consequence for the cancer. So far for the first patient, there were 7 mutations identified through sequencing. So next, we wanted to validate these mutations through a custom ultraseq panel. So we designed the ultraseq panel to detect these 7 mutations. And here we have the results of the ultraseq panel. The samples were run with a set of 28 wild-type samples to establish a baseline. And all of these 7 mutations were multiplexed into a single reaction. And as you can see, the results match the sequencing, and no additional low-level heterogeneity was discovered. So you can see here on the smaller chart, the blue dots represent the wild-type samples, the red dots represent the mutations. So in some of the cases, we did not detect the PIK3CA and the MAGI2 mutations in some of the samples, but in other cases, the mutations are present and at a high intensity. We think that we probably did not pick up any tumor heterogeneity most likely because it was too low, probably less than 0.1%, and hence could not be detected. So next we used digital droplet PCR and ultraseq to look at the results or validate the results in circulating cell-free DNA. The samples were run in duplicate. With only 15 microliters of plasma available, we had about 750 copies of DNA available per reaction. So we were able to find the mutations in BRAF, RB1, TP53, and MLL2. For the 3 of these genes, we were able to detect both replicates, whereas for MLL2, we were only able to detect one of the replicates. We think we missed the other one, possibly due to sampling error. You can see the data point is just very close to the whisker, so maybe due to low input DNA. The other 3 genes, which is PIK3CA, did not have any mutations present in the plasma DNA. So to summarize the results of this one patient study so far, the sequencing results for the tumor biopsies match with the results from Ultraseq. Of the 7 mutations detected, Ultraseq was able to detect 4 of the mutations in plasma as well, indicating that and the results showed that the intensities for these mutations were pretty low, indicating that these mutations are all below 1%. So this could explain why we only saw 1 mutation present in one of the replicates for MLL2. So it's interesting here that the mutation profile in plasma matches that of the lymph node. What is really neat about the study, even though we only have data for 1 patient so far, is that we were able to create a patient-specific custom Ultraseq panel where we could go from design to results within 5 days, just 5 days. This panel can be used to monitor the patient's disease status non-invasively. The sequencing of the patient for the tumor samples took about 4 to 6 weeks. So from the study, you've seen now how you can discover mutations through the various next-generation sequencing technologies and then use the mass array system to confirm and validate these mutations. Now, we will soon have a design tool where our users can design their own custom Ultraseq panels to detect and validate low-frequency mutations. So this would complement the panels that have already been created and are on the market from Agena. So by creating these patient-specific panels, this allows you to move towards the ultimate goal of personalized medicine, where you can discover mutations through next-gen sequencing and then develop custom panels specific to each patient to detect driver or disruptive mutations from circulating cell-free DNA non-invasively and provide real-time treatment options. So through this presentation, I have introduced to you Agena's Ultraseq technology, which can identify somatic mutations present at very low frequencies, down to 1.1%. We have proved the performance of this panel with cell-free plasma-derived tumor DNA. We also evaluated it against ARMS PCR, where Ultraseq was found to have an outstanding sensitivity, detecting an additional 21% mutations that were missed by ARMS. The amazing flexibility of this technology allows you to create patient-specific custom panels and test them within 5 days. No other technology gives you this level of flexibility combined with a quick turnaround time. So in closing, Agena's mass array system provides you with high-quality data where you can precisely detect and quantify extremely low-frequency variants. The technology can be used for not only the detection of somatic mutations, but also genotyping of germline SNPs, indels, determining copy number variants, quantifying expression and methylation profiles, as well as detecting microbes. The flexibility of the platform allows you to easily design assays for your biomarkers of interest. This is a very cost-effective platform capable of high throughput and is a proven technology with over 1,500 publications. We would like to thank our collaborators from the London Research Institute, AstraZeneca, and Sequenome. And I think with that, completed the presentation. And I think, Elizabeth, we can get into a few questions.
Thank you very much, Divya. We have several that have come in, and there's still time. If you have a question for Divya, please chat it in using the chat panel, and just send it to all panelists. The first question is about something that they may have missed during your presentation. On the gene panel, they weren't able to see the BRCA gene. Was it included?
Let me pull up that slide so you can see exactly the mutations present in our current panel. Here we go. We actually do not have the BRCA gene on our panel, but again, we offer custom panel design. So either we can design a panel for you containing the BRCA gene, or it can also, we'll soon have a software application available where users can develop your own custom panels.
Okay, thank you. And a question about a definition. When you say ARMS PCR, are you referring to application refractory mutation system?
Yes, that is the technology being referred to.
Okay, they just wanted some clarification there. Another question, what's the lowest level of mutation that you can detect?
With our panel, we can get down to 0.1% mutation detection.
We have a question from Rawia. If you're using an already made panel, how can you make the study novel?
So we can offer custom panels. So based on Rawia's study, we can generate a panel that is specific to the mutations that they are interested in. Current offering, our Oncogene Ultraseq panel, does not contain the mutations that they're interested in. We can develop a custom panel for them.
All right, perfect. We also seem to have a request for another webinar from you. They ask, do you expect to do a webinar with iSeq or CNV and QGE? I think we have a few more webinars planned for the new year, and I'm not sure about the topics, but I will definitely pass on the interest for iSeq, CNV, and QGE. Okay, perfect. Christiane asks, are you planning to validate this platform for diagnostic use? Currently, the panel itself is a research use only panel. The instrumentation, the mass array system, is a 510K approved instrument, but we are open and would be interested in speaking with collaborators who might be interested in taking this for diagnostic uses. All right, perfect. Mark asks, can mass array cover copy number variations or translocations? Yes, we can do both. The mass array system, we can design assays to determine copy number variations. We currently have a few commercially available panels. For instance, in the pharmacogenomics area, we're looking at copy number variations in 2D6, for example. And we also have panels for translocations as well as fusions, for instance, EML4 ALK and other lung fusion panels as well. Thank you. A question from Maureen. How much plasma is required to provide the 40 nanogram DNA? So typically with the 10 mils of blood gives you about 4 mils of plasma, which gives you around 6,000 copies of DNA. And so for the 40 nanograms, you need about a 10 mil blood draw, which is 4 mils of plasma. And a question from David. Is there a target amplification step during mutant detection prior to the capture steps? We have an initial amplification where we, let's go here, where we're amplifying both the mutant and the wild type signal. And then in the single base extension, we are doing a mutant specific extension reaction, and we're only capturing the mutant alleles, whereas the wild type allele is washed away. So just initial standard PCR followed by the extension with the mutant specific streptavidin bead labeled nucleotides for the mutant alleles. All right, Maura asks, what's the maximum plex reachable with Ultraseq? So currently, for instance, our current panel with the 26 mutations, this has been multiplexed into 4 reactions. And this is based on the mutant allele because we want to multiplex
mutations that have the same mutant allele into the same reaction. So I would say you could multiplex up to, just like for our other somatic mutation assays, maybe up to 10 to 12 mutations in a single reaction.
All right. Piotr asks, what about working with fusion genes?
With fusion genes, yes, we have some panels already developed looking at translocations and fusions in the ROS1 gene as well as EML4 and ALK. As long as we know the breakpoints, we are able to design assays for them. I just wanted to clarify about that question on using this diagnostically. I mentioned that the instrument is 510K approved, and it is 510K approved with a factor 2, factor 5 assay. We have that currently for the instrument available. If they wanted to take it to a diagnostic test, we would have to develop the required testing and panel along with the instrument.
Okay, and we have a related question there to your clarification. This person says, I'm planning to develop a panel to detect breast cancer. Is it possible to use the UltraSeq oncogene panel? And if so, how would you approach that?
We have the current UltraSeq panel, and they can certainly use that. If they wanted to add some additional breast cancer-specific mutations, we can certainly work with them to help develop a custom panel that would meet their needs.
Perfect. Another question there, flowing in fairly steadily here now. Which cfDNA extraction method have you used or do you recommend?
So we have used the extraction kit from Agena. It is the high-amp, I think, let me make sure I get the name correct, the high-amp circulating nucleic acid kit. So that's the one we have currently used.
All right, and we have another question that just came in that's related. Melinda asks, what platform do you use to extract DNA from plasma?
It would be the same, the high-amp circulating nucleic acid kit. We just use a kit to extract out. As far as I know, I don't think we are using any automation at this point.
Another question, what is the minimal and maximum size of the custom panels that you can make?
This is the best part, the flexibility of the platform. We can, if you just are interested in a single mutation, we can make a panel with just a single mutation all the way to hundreds of mutations. It just depends on what is required. So very flexible assay design software and platform. We don't have any fixed sizes or combinations of assays and samples. So you can put in as many mutations as you would like. We will do the design to multiplex the assays, and you can run them on as many samples as required.
And a question from Anne. Is there a difference in the sensitivity limit for different substitutions?
No, at least from our evaluations so far, we have been able to detect all the 4 different alleles successfully.
Perfect. And a question from Tiling. How do you compare your method's accuracy with regular DNA sequencing in detecting mutations?
We have been, as we saw in the case study, we were able to detect all the mutations that were found in sequencing, but we also understand that for sequencing and for detecting these low-frequency mutations, the current sequencing technologies don't have that high level of sensitivity to detect mutations present in below 3% or down to the 0.1% that we can for detecting mutations present in circulating cell-free DNA as well as our plasma. For validating some of our findings, we used other platforms such as ARMS and digital droplet PCR to be able to achieve this level of sensitivity.
All right, and a question from Cedric. When you're looking at multiple genes, is it a multiplex analysis or do you have to analyze each gene individually, which would then require more material?
This is a multiplexed analysis. So a single reaction can have mutations from multiple genes in the same reaction. So that way you don't need additional material. So with the 40 nanograms of DNA, we are able to assay all these 26 mutations in these various genes. So it's very low amount of DNA.
Oh, sorry about the interruption. Monica asks, has anyone tried this technique to detect somatic mutations in the brain?
Not as yet. This is a newly launched panel, and we would be very excited if people would be interested in looking at it in various tissues. So right now we've only looked at plasma and focused on somatic mutations that may be present in plasma, but it would be very interesting to see what we would find in the brain.
Okay, perfect. Sen asks, besides F1174L, can UltraSeq detect other ALK mutations?
Yes, for this current panel, we chose this particular mutation, but if you were interested in other ALK mutations, we can certainly develop a custom panel for that.
All right, getting down to the last few questions here. What's the advantage of cell-free compared to CTCs?
So with cell-free DNA, typically we have higher yield. So the circulating tumor cells require a cell sorting technology to extract out the cells, which can be a bit more expensive, and overall the yield is diminished. One benefit of CTCs is that you have the whole cancerous genome intact, but the overall yield is lower. Whereas with cell-free DNA, you typically have a lot more DNA just available in the plasma. It's easier to extract and less expensive. That would be the benefit.
One last look over all of the questions here. It looks like that's all of them.
Great.
So I would very much like to thank you, Divya, for your wonderful presentation and answering all those questions as well. I'd like to thank Agena Bioscience for sponsoring today's web symposium. And most of all, I'd like to thank those of you who took an hour out of your day to come and join us. And I hope that you were able to get information that will solve some of your problems or make your job easier. So with that, I will wish you all a good day or a good evening. I look forward to seeing you at future CHI Global Web Symposia. Thank you, and bye-bye.
Thanks, everyone, for attending, and thank you, Elizabeth, for moderating this session.
You are very welcome.